Right, good morning, everyone. Thank you very much. I'll just start by acknowledging the traditional owners of the lands on which we meet. In particular, pay our respects to elders past, present, emerging, and acknowledge that we're on Bindal country, and the remaining part of this workshop is going to move to Magnetic Island. So also want to pay our deep respects to the Wulgaroo Kabar people. So it gives me great pleasure today to launch a workshop that we're hosting for the next week. And as a consequence, we've got a number of visitors who I welcome to the centre, and very, very happy to reconnect with some of these people who we've been working with for a long time. So not only have we got Shaun Wilson here, who's going to give the seminar, but we've also got Professor Michael Berriman from KAUST. We've got Jordan and Claire here. Oh, Claire. Also from Department of Biodiversity, Conservation and Attractions in West Australia. And we've also got Laura Richardson from Bangor, and a number of other local people who are also going to be contributing to the workshop, including Mike Emsley from AIMS. So if you want to meet with any of these people, there's the opportunity to have a one-on-one tomorrow afternoon between 3 and 5, or please feel free to come and join us at the Riverside, or Riverview, I think it's officially called, Tavern in Douglas from 5 p.m. tomorrow. So on to the main reason you're here today. It gives me my great pleasure to introduce Dr. Shaun Wilson, who's the Principal Research Scientist and Acting Director, is it, of the Marine Science Program in the Department of Biodiversity, Conservation and Attractions for the West Australian Government. Shaun is a JCU alumni. I had the great privilege of going through undergraduate honours and my PhD alongside Shaun. He did his PhD under the supervision of Dr. David Bellwood. And since that time, he's produced an exceptional body of work focused on things like habitat associations, diets, and roles of reef fish. And in particular, he's forged a really impressive body of work related to the effects of environmental disturbances and other anthropogenic pressures and the changes in not only coral reef systems, but also increasingly subtropical marine systems. And that's sort of the reason, that's part of the focus of the workshop that we're going to be hosting. Can you please mute if you're online? We'll open up for questions at the end. So I can tell you lots about Shaun, obviously, but I won't embarrass him, and I won't cut into the time that he's got to talk to you today. But I do encourage you, if you don't know Shaun and the other visitors, I suggest you make the time to try and meet with them tomorrow afternoon. Thanks very much. Over to you, Shaun.
Great. Thank you very much, Morgan. Thanks to you and also to the Centre of Excellence for hosting us here and having the workshop. I thought I'd also just take the opportunity to introduce Jordan Goetzer, who's here with us today from Marine Science Program. So Jordan has been doing a lot of work and has expertise in using grubs and doves, and is one of our resident fish ecologists within the Marine Science Program. Recently published a paper using grubs around Australia to try and understand the effects of marine reserves around the continent. And also Claire Ross, who's up the back, who many of you may know because she did her PhD here at the Centre of Excellence, although based in WA with Malcolm McCulloch, and did a lot of work on coral growth and a lot of things like accretion of corals and calcification, and is now in charge of the monitoring program for our corals as well. So has recently just published a paper looking at the distribution of corals in the southern part of the state within the different marine parks. So they're the two topic areas that, if you're interested in following up with those two guys, it'd be worth sort of talking to them about. So today, I was going to talk about tropical macroalgae or seaweeds, and this is a really, I guess, often a prominent part of the natural sort of tropical seascape, and it often covers extensive areas of the tropical seascape. So this is a picture that I think demonstrates that quite nicely from a reef in the Seychelles. those macroalgal fields, or they often sort of break up seasonally, and they form these massive sort of rafts that provide habitat for a number of fish and vertebrates, and even things like little turtles. They then can sometimes wash ashore and form wrack on the shoreline that nourishes that shoreline, and again provides a lot of habitat for invertebrates in particular in that area. But it's also an area of controversy for a lot of places around the world because it's choking up tourist beaches. So a lot of interest in that area. And then the focal area really that I'm going to talk about today is the use of these macroalgal habitats by fish in particular. And so there is a reasonable amount of literature that has talked about that in the past. And it's often sort of focused on differences between corals and macroalgal habitats, and often given negative connotation in terms of regime shifts. But I think we're beginning to appreciate that there's a lot of positive aspects as well in terms of how these habitats contribute to the wider biodiversity of the system across the seascape. So most of the work that I'm going to present in the first part comes from the talk comes from Ningaloo. And Ningaloo, for those of you who are unfamiliar with this particular reef system, is a long fringing reef. It goes for about 260 kilometres along the west coast of Australia. Although we appreciate it as a coral reef system, hyperspectral mapping, admittedly about 10 or more than 10 years ago, finds that only about 6 square kilometres is actual coral habitat. And much more of that habitat, about they estimate 39 square kilometres, is actually macroalgal or turf habitat. And particularly in this sort of lagoon area here, there's these large meadows of canopy forming macroalgae dominated by things like sargassum in particular. Now, our initial work here was a project, was a WAMSI-led project, and it was led by Marcel Petchensky from AIMS, who a lot of you may also know. And we were doing surveys both in the macroalgal and the coral reef habitat to try and identify what are some of the key habitats important for recruitment of fish and what are the juvenile habitats that they use. And so this plot up the top here shows the probability of about 50 different species of finding them in either reef habitat in orange or macroalgal habitat. And I guess most people would expect that there's a high probability of finding a lot of these species in a coral reef environment, but somewhat surprisingly, or was surprising for us at the time anyway, was that there was a lot of fish that were almost exclusively or predominantly found, and a much higher probability of being found in the macroalgal fields or meadows. And this included things like some of the acanthurids and the mullets or the goatfish, and probably most interestingly, a lot of the lephrinids. And that's interesting because the lephrinids are an iconic species, particularly nebulosus or the spangled emperor at Ningaloo, and really important for the recreational fishery there. So when we look at the sort of the composition a little bit more of the juvenile fish in these two habitats, and here we've got our different sites along the coast of Ningaloo. Some of these just got macroalgal, or sorry, coral, so the purple ones, and some of the areas are paired with a macroalgal site, and they're in green or grey, depending on what your site's like. But what we found was that typically the number of species and also the abundance of fish that we're seeing in the macroalgal areas compared to the coral is much lower. And that's generally, you notice it really when we're doing the comparison between sites, macroalgal and coral within the same sort of region. Somewhat more interestingly, though, is the number of rare species that we see. So we classified rare species here as those that represented less than 0.5% of the abundance in either macroalgae or coral habitats. We find that very comparable in terms of coral and macroalgae. And then when we do some of the phylogenetic or taxonomic distinctness type measures of diversity, we find that in many instances, the diversity measure is actually higher in the macroalgal habitat. And I think that's because on the coral reefs, the juvenile assemblage is really dominated by a lot of the labroid type fish, so pomacentrids, the scarabs, and the labrids themselves. Whereas when we move into the macroalgae, we've got much more diverse or different types of families. So we've got mulids, we've got the acanthurids, we've got the siganids as well. So a lot different, and as well, we're also getting a lot of those labrids. So I guess the point that I wanted to make with this particular slide was that yes, the abundance and species richness may be low, but there's a unique component of that assemblage that is contributing to the overall diversity of the fish across the whole seascape when we look at it at a broader picture. We also found that there's, as the fish recruit into this type of habitat, there's evidence that they then sometimes can move back onto the reef. So this is work that we did with the three left-rhinid species, and all three of them recruit, or I shouldn't say recruit, but as juveniles, we're finding them pretty much exclusively within the macroalgal habitat. And then with this particular species, the yellowtail or left-rhinid Atkinsoni, at about 15 to 20 centimetres, they start to migrate and move onto the reef. And when they're really large, we don't see them at all in the macroalgal habitat. Nebulosus or the spangled emperor, again, about 15, 20 centimetres, they start to appear more prominent on the reef. But then the really big ones, we're seeing just as many of those on the reef as we're seeing in the macroalgal habitat. So some of them obviously stay within that area. And then the smallest of these species, variegatus or slender left-rhinid sometimes it's called, pretty much stays within that habitat all of its life, it seems. So we've got examples here of species that have ontogenetic shifts from macroalgae onto the reef, some that may move back and forwards on the reef, and some that pretty much stay within the macroalgal fields throughout their whole post-settlement life history. We also found at Ningaloo that not all macroalgal fields are equal. So some places we went to and there was literally clouds of juvenile fish, and some absolutely nothing. So this somewhat crazy sort of tile plot here just shows abundance of juvenile fish that we saw and how that changed both among the different sites and across about 6 years of survey. And you can see that there's considerable variation among the sites. So darker sites are where abundance is higher, but also even among years. And so it got us thinking about what are some of the reasons that we see that type of variation. Obviously, some of it relates to differences in the habitat, particularly the habitat provided by the macroalgae. And so we started looking at things like the number of holdfasts to give an idea of the density of the macroalgae. We measured the canopy height, both as it sits in the water, but also as it extends and pull it up as high as it will go. And then overall cover, and also the community composition of the macroalgae as well, but I'm not going to present any of that today. Also, post-recruitment processes obviously important here, so things like predation. But then the temporal shifts, potentially changes in annual supply rates. And I'll just talk briefly about each of those. So in terms of spatial variation, if we looked at coarse measures of diversity, things like species richness, and we compared things like the canopy height, number of predators and cover, we found that the overall density, so the number of holdfasts, was a good predictor of species richness. So typically low density was associated with low species richness and vice versa. And That wasn't really consistent though among species, as you might expect. So some of the species like Kileo inermis and Bagiensis, yep, strong positive relationship with density. But others, there's actually negative relationships and some really no relationship at all. And then when we looked at things like cover and height, we find that some of these species, they're positively associated with things like height, so like the Spatial juventa, some of them negatively associated with height and cover. So the point here is that each of these species seem to have more specific sort of habitat requirements associated with the macroalgae. And we can go even further than that, and we find that, and I haven't got a slide to demonstrate this, but some of the species associate with even particular genera of the macroalgae. So sometimes Sargassum or sometimes Sargassopsis as well. So the other, I guess, really interesting thing about macroalgae, and in the tropics, and I alluded to it a little bit earlier, is that it goes through these seasonal shifts in its structure and abundance. So here's some work that we did very early at Ningaloo. The grey sort of outline shows the biomass of Sargassum and how that changes over a period of about 18 months. And at these two sites, both in the north and the south of Ningaloo, we tended to get high biomass within the summer months, which coincidentally is also when the fish recruitment is occurring, or we're getting most of the fish recruitment. Then it breaks off, forms those rafts that I showed you in the first slide, and we get some of the understory sometimes becoming more prominent, which is structurally things, particularly things like Lavophora for fish, probably structurally maybe not quite as complex. And so I guess the question is, with this change in habitat with winter, some of the early things that were posed to us as a group, what happens to those fish that are recruiting into that habitat? So some of them obviously recruit, or they undertake ontogenic shifts onto the adjacent coral reef. But some of what happens about those fish that live within the macroalgal habitat for their entire post-settlement life history? So part of the answer, I think, here relates to the actual structure of the macroalgae and how that differs among sites. So some places we're seeing that, as you'd expect, the density and height and the cover is really high in the summer months, so they're coloured in brown, and lower in the winter. But then we have these weird situations where sometimes the density or the height is actually higher, or the cover is actually higher during the winter. And so it infers that some of these, there's not a consistent pattern in terms of seasonal change among the sites. And so these sites potentially are really important for providing ongoing structure and complexity for the fish that persist over those winter months. So this is work that was done by an honours student supervised by Chris Fulton and myself, Isis Lim, and she did surveys in the summer and the winter at Ningaloo and found that the best predictor of changes in Leptoscarus bagiensis, so this is one of those species that we find always in the macroalgae, we never really see it at all on the actual coral structure. Best predictors of change in their abundance over that summer-winter period is either changes in seaweed cover or the canopy height. So when there's a decline in both of those, we see a decline in the abundance of those fish. And when it remains stable, or there's actually a slight increase in the summer months, then we actually see a slight increase in the abundance of Leptoscarus as well. So from a conservation perspective, understanding, I guess, what it is about those sites that is environmentally important becomes sort of critical, because they're the ones that these macroalgal fields are the ones that are going to help these populations persist over that winter period. The other temporal dynamic that I mentioned was changes in supply rate of recruits. And as I'm sure most of you are aware, the supply rate of recruits changes from year to year. And that's certainly what we observed at Ningaloo. Some years here in 2010, we got virtually nothing on a transect, and then the following year we were getting huge numbers, like 40 juvenile fish per transect. Transect is 30 meters by 1 meter, so it's not a huge area. And that got us thinking about some of the other work that's been done in the temperate areas of Ningaloo, particularly by fisheries, and understanding how the supply rates of things like the western rock lobster are really closely tied to enzo events. And so they've actually looked to see that things like the Pirulus for the western rock lobster, their supply rates correlate really nicely with things like the Southern Oscillation Index. And that's become an important part of the management of that fishery. So we thought we'd see how that relates to our juvenile fish at Ningaloo. And somewhat surprisingly, for me at least, the relationship was incredibly strong. So what we were finding was that during La Nina years, so in WA, La Nina years are associated with a strengthening of the Lewin current that runs down poleward direction from north to south along the Ningaloo coast, and also warmer temperatures associated with it. When we have those conditions in La Nina, we tended to get more juvenile fish, in this case particularly reliants, and El Nino conditions were associated with really low counts of juvenile fish. And that's when we had weaker current and typically lower temperatures, sea surface temperatures. This is really, I guess, potentially important, particularly for things like reliant, for understanding what that stock's going to look like in future years, but also for our monitoring plan in terms of understanding what natural fluctuations in the abundance of fish that we're monitoring is going to look like in the future. So that sort of brings me to the end of the section that I was going to talk about Ningaloo. But one of the questions that I often got when I was presenting Ningaloo data was, that's all very nice, but Ningaloo is a pretty unique system. How does that relate to the rest of the world? Unfortunately, while we've been doing this work, and also in other parts, even before we were doing this work, there's a number of people that have been doing similar sort of work, including people in this room, like Andy and his students, on reefs in other parts of the world. So Chris Fulton really led the charge in this area, but did a search of the literature for surveys that had been done in macroalgal areas to understand relationships between macroalgae and fish. And we also, at the Indo-Pacific Fish Conference in Tahiti, and also at the Australian Society of Fish Biology that was in Albany, put out a call to people to say if they had unpublished data that they were willing to share that might provide information on habitat associations of fish in macroalgal, tropical macroalgal areas. We ended up with 24 independent studies at 17 locations around the world, and information on 633 species. We got also a number of studies, so 12 where we were able to compare fish in macroalgae and coral, and 7 where we were able to look at macroalgae in seagrass versus seagrass, I should say. And the first comparison using that data that was collected on both coral and macroalgal reefs. Here in green, you see that there's about 17 on those studies, or among those studies, I should say, about 17% of the species were unique to macroalgal reefs. And then about 39, I think is the actual number for coral reefs, and the rest were seen in both habitats. Those numbers somewhat switched around when we were comparing macroalgal and seagrass habitat. So when we did that comparison, about 40% of the species that were seen in studies were unique to macroalgae, only about 20% to seagrass habitats, and then 40% were found on both. So it somewhat confirms our work from Ningaloo that there is a unique component of the fish assemblage across that tropical seascape that is associated with macroalgal habitats. We're fortunate that a number of the studies also collected information on adults and juveniles. So in this plot, we've got the proportional abundance across the studies for adults that we saw, and on the x-axis is proportional abundance of juveniles. And the majority of species are really in this bottom corner, which infers that they're predominantly associated with coral, both as adults and juveniles. But there's, again, a number of species that spend pretty much most of their life post-settlement in macroalgal habitats. And then this quadrat down here, where there's 9 species, is indicative of those that would recruit into a macroalgal habitat and then migrate into coral. And then there's these ones here that either recruit into coral and migrate into macroalgae, or spend a lot of their time actually foraging in the macroalgal habitat. The other thing that we've done here is to plot with regards to trophic level. So I guess the interesting thing I find here is that the species that we're finding in macroalgal habitat are often in the lower trophic levels, or those mid-tier sort of carnivores. There's not a lot of these sort of higher trophic levels that are greater than 4, but they do occur occasionally. So there is some predation occurring, obviously, within those habitats or within the macroalgal area. And I guess what this is confirming is similar to what we showed with the left-reliant study, is that there's some species that are in macroalgae all their life, some that and some that undertake ontogenetic shifts in habitat use. We also wanted to look at habitat specialization. So a number of the studies also provided data on macroalgal cover, and we were able to look at abundance across within those studies and how that changed with cover, and just basically get a correlation coefficient. We then averaged those across the studies and weighted them based on the area that was surveyed. And so areas that, or species here that have got crosshatches on them, are those where we get significant differences from 0, so that there is a slope, and that's inferring that there is a relationship between their abundance and increasing algal cover, and I guess an inference that there is a level of specialization on that habitat. Again, we see that for some species that specialization occurs both in the adult and the juvenile stage, and for others that's really only occurring in the juvenile stage. Interestingly, that's Atkinsoni again, the yellowtail left-rhinid. And we're also getting a number of adult species which they occur in macroalgae, but there's actually a negative relationship in terms of their cover. So I guess that means that they're there, but they really only like a little bit of macroalgae. The other thing I just wanted to point out, because it sort of leads on to the next section, is a number of these species which have got asterisks on them, we've highlighted as being potentially important for local fisheries. And I guess what we really wanted to do is get a better understanding. We know, I hope I've convinced you that macroalgae is an important habitat for a lot of tropical fish, but how important is some of the ecosystem services associated with macroalgae? So in particular, one of the obvious ones is fisheries. We get a little bit of an idea of this from some work that James Robinson did in the Seychelles. And a lot of you may be aware that in the Seychelles in 1998, there's mass bleaching that occurred there. and unfortunately they lost variable among sites, but somewhere between 70 and 99% of live coral. And in on some of those areas, that was replaced by large stands of macroalgae, things like sargassum. And so James worked very closely with Seychelles Fishing Authority, which had been collecting information on catch since 1994 and continue to do so, but just presented here as 1994 to 2015. And this was for two types of trap fishery. One of them is the top row is the fixed trap fishery. So that's where these traps up the top here are left in the water, usually for 1 to 3 days. And then the other one is the active trap. So same sort of trap, but they're really only left in the water for a number of hours. What we found interesting was that despite this massive change in habitat that occurred in 1998, the fishery, and in particular these ones here, which are the everything combined, remained relatively stable, or in terms of catch per unit effort, I should say, or it actually increased. And it's really obvious in the active trap fishery, particularly for things like steganids and lephrinids. And both steganids and lephrinids tend to have a really high dependence on macroalgae. Lephrinids often during the early life history, and steganids early and also later life history stages. And we followed this up, investigated a little bit further. So since 2005, Nick Graham and I have been visiting 21 different sites in the Seychelles and tracking how they change over time. So of those 21 sites, 12 have slowly been recovering so that they become more like they were pre-98 and becoming again dominated by coral. And then another 9 of those sites were completely different and now and continue to be dominated by this canopy forming macroalgae, often sargassum, but sometimes terminaria. And what we found is when we look at the UVC data that Nick had collected over the years and classify the fish as either primary or important targets based on the information that we get from Seychelles Fisheries. In general, we're seeing as the macroalgae has changed or increased in time on those macroalgal reefs, we've had an increase in the water or availability of these primary target species, and to some extent or lesser extent, also important target species. So again, I think that gives a bit of a hint of the importance of macroalgae in fisheries, but we wanted to try and make some more general statements using information from the literature. So again, we went back and had a look at what we can find in terms of species-specific catch composition data, which I don't know if any of you have tried to find that, but it's actually pretty difficult to find. There's not a lot of it out there. But we were fortunate enough that we went back to our collaborators that had provided habitat information. They were able to provide some information that wasn't published. There was a little bit in the literature. We ended up with 91 locations from 28 countries that we had data. And what we did was we used the habitat data that we collected, both for the juveniles and the adults. And for each of the species in the catch, we estimated the proportion that they're associated with macroalgae based on their abundance in coral and macroalgal habitat. And it's worth stressing that this is just considering coral and macroalgal habitat. And then multiplied that by how much of the catch they represented and divided it by the total catch. And that gave us a broad sort of indication of the importance of macroalgal habitat to the overall fishery. What we found is, and it's this top figure here that I want you to look at first, is that overall, of all the data that we had, about 25% of the catch is supported by macroalgal habitat. That varies among locations. So some places like in the Philippines, and also in Kenya and in the Pilbara coast along WA, it's regularly well above 50%. But then there's other places in the world on the other side of the ledger. A lot of those are Pacific island nations where it's regularly much lower than 10%. So it's quite variably location-wise. We also looked at it in terms of different fisheries. So the recreational and subsistence fishery tended to be a little bit higher than things like the commercial and artisanal fisheries and experimental data that we had. And we also saw that there was slightly higher representation with some of the gears, particularly spear and trap fisheries. Next, we used the same approach, but to try and get an idea of how these estimates may differ if we look at reliance on particular habitat, coral or macroalgae. We only included in our calculations, first of all, those species that their overall abundance was predominantly in either coral or macroalgal habitat. And here, we took an average of both juvenile and adult habitat use. So I appreciate that some species will have very specific habitat associations of juveniles, but that's not necessarily captured here. What we found was that when we did that, the overall contribution of macroalgae was much lower, so less than 10% of the fishery, and much more dominated by those species that rely on coral reefs. We then took it a couple of steps further. So now we only, in this one, included fish that 75% of all the reportings occurred in one habitat or another, then 90%, then 95%. And finally, those fish we only included species that were only ever found on coral or macroalgal habitat. And I guess this is the thing that I found quite interesting from this particular figure, is this last point here. So there, even when we look at the coral, what we call coral reef species, they only represent 10% or less of the overall fishery. And I guess the thing that's important for me here is that it's probably, a conglomeration of different habitats. Here we're only talking about macroalgae and coral, but obviously there's other habitats within that whole seascape which are really important for supporting the fishery. It's not just the corals or the macroalgae. And so if we're managing that system in terms of ecosystem management, we need to be looking at it from an ecosystem perspective, not just corals or macroalgae. We're also interested in how the fishery may change in terms of its vulnerability to fishing. And for this, we used the species values that William Chung developed, and using his calculations, we assessed the vulnerability of the catch and then plotted that against the proportion of the catch attributable to macroalgae. And there was a general sort of downward trend. So the interpretation here is that as the fishery becomes more dependent on macroalgae, it actually becomes less vulnerable to overfishing. And I think this is really probably driven by the fact that a lot of these points down here are fisheries that are characterized by large amounts of things like sagannets, which have very, very rapid growth rates and fast turnover, and potentially, maybe a little bit more sustainable than some of the fish that are much larger body, have slower maturation rates and life. However, on the counter to that, if we look at the biodiversity of the catch, and that's measured here just as Simpson's index, as the fishery becomes more and more dependent on macroalgae, there's fewer and fewer species associated with the fishery. And so that means that the fishery is obviously at greater risk of natural fluctuations in abundance of those stocks. The other important point here, and it just is that the actual highest diversity in the catch is not when we have 100% coral dependence, but it's at about 20%. So again, emphasising the importance of having and managing different habitats across the seascape for the fishery. So in conclusion, I guess there's a few sort of points I wanted to make. One is the importance of macroalgal fields within the seascape, both in terms of providing or contributing to the overall diversity of the fish assemblage, but also to the fisheries, which we're estimating is about, they're contributing about 24%. It's probably a little bit of an overestimate in some instances, because remember, we're only comparing coral and macroalgae in those models. The importance of macroalgae often relates to things like the diversity of the canopy structure, so its height and its density, and particularly for species that are always in that habitat, they require sites that maintain that structure over the winter months. And in terms of the fishery, it may be that those sites or fisheries that are heavily dependent on macroalgae have lower vulnerability to overfishing, but they're also vulnerable because there's lower diversity within those fisheries. So there's a little bit of to and fro in that sense. And with that, I'd just like to acknowledge particularly the traditional owners of Ningaloo, where a lot of our work has been done. And my colleagues at Department of Biodiversity, Conservation and Attractions and AIMS, who've been instrumental in collecting the data at Ningaloo over a number of years. Lancaster University, Nick Graham and James. Nick's led the charge really at the work that we've done in Seychelles for a number of years, and I'm sure you're all aware of some of the fantastic work that he's done there. And also our colleagues in the Seychelles, particularly Seychelles Fishing Authority, that has supported that work. All of the people that provided data, particularly for the Habitat Association work. And then finally, once again, thanks to the Centre of Excellence and to you, Morgan, for hosting the workshop. Thanks, guys.